So today we will be talking about infectious diseases, continuing on our little, multiple topic discussion for the rest of this semester. Today we'll be focusing on the nervous system, cardiovascular system, and the respiratory system. So it's going to be kind of a lot. The good thing is a lot of these kind of go hand in hand. That's why I've kind of combined the lectures. Also, it makes for one less lecture, which gives us a review session at the end. So it's worthwhile doing. All right, so a little bit about the nervous system. It's basically composed of two different components, the central nervous system and the peripheral nervous system. So the central nervous system is the spinal cord and the brain. The peripheral nervous system are all the other nerves throughout the body. So the peripheral nervous system, what innervates all muscles, tissues throughout the body. And the central nervous system is the spine and the brain. Pretty basic. Should be somewhat review since for anatomy and physiology. Nervous system defenses. So the unique thing, especially about the central nervous system, is that it is an immunoprivileged site, meaning there are no immune cells in this region. So what that means is if there's an infection, you're SOL. It's a good thing that it's an immunoprivileged site because you do not want a large activation or immune response of activation of an immune response in the brain. And this is primarily because anytime you have immune cells that are activated, they're releasing cytokines. Cytokines result in inflammation. Now, inflammation in the periphery is fine, but as soon as you get inflammation in the brain, you run into a lot of problems. The biggest problem is the skull. The brain is completely surrounded with a bone. So if there's any pressure buildup in the brain, it's got nowhere to go because the skull kind of sequesters it in there. So instead it turns in on itself and pushes on the brain. So whenever there's inflammation in the central nervous system, it generally causes neuronal death or damage. So the biggest thing you'll see with all of these infections that we'll be talking about today is either death, seizure, coma, or major neurological sequelae. And by major neurological sequelae, that means exactly that. The reason it's such a generic term is because depending on where the inflammation occurs in the brain, that depends on, or that determines what type of sequelae you're going to get. So for example, if you get major inflammation in the occipital part of the brain, you're probably going to have issues seeing. So wherever you get the pressure, wherever you get the neuronal death, those are the sequelae that come with it. So as you can imagine, having major neurological sequelae in the brain is probably a bad thing. So a lot of these disorders and diseases are pretty nasty things to get. So since it's an immunoprivileged site, the biggest defense in the brain is the fact that it is sequestered from the rest of the body. So right around here in the brain stem, you have something called a blood-brain barrier, which essentially is a tight junction formation of cells that block anything that could be found in the blood or in the rest of the body from penetrating through the brain. It usually works extremely well, but pathogens that like to infect the brain have figured out ways around this. And usually the easiest way around this is just to go through the cells of the blood-brain barrier. And they do this fairly well, usually by burrowing. Since it is a closed system, it has its own fluid associated with it that never comes into contact with blood. This fluid is called cerebral spinal fluid. Essentially keeps the brain bathed, moist, and gives it nutrients to a certain degree. There's also a blood flow, but the blood flow is passed through a filter before it makes its way into the brain. So this filters out any unwanted organisms. The meninges, so the meninges is a thin connective layer tissue surrounding the brain, works primarily as a way to kind of keep the brain enclosed with something, a nice little thin protective membrane. However, it is a perfect place for pathogens to infect. So most of the diseases we'll be talking about are meningitis, which are infections of the meninges. You'll find the meninges all throughout the cerebral or the central nervous system. around the spine, and also surrounding the brain. And it's an immunologically privileged site, like I mentioned before. There are some specialized cells in the brain, like astroglia or microglia, which act kind of like immune cells, but they have a lot of their functions taken away. So they do phagocytose, but very limitedly. And when they phagocytose, they do not become activated like a normal immune cell would. So they do not cause inflammation. However, if there is an infection in the brain, enough of these cells will come into contact with it. They will slightly activate, and this is what results in the inflammation that can occur. But generally, the inflammation is kept to a minimum. But even just a little bit of it is pretty serious stuff. So normal biota of the nervous system? None. It's sterile site. And I remember before I mentioned that there were, a list of sterile sites in the body, most of which, tend to have passing microbes occasionally. The brain is the one exception to this. It is always sterile. If it is not sterile, you have an infection. So one of the easier diagnostic tests for a brain infection is to do a spinal tap. Pull out some CSF, cerebral spinal fluid, either culture it, PCR it, look for organisms, and if you find anything, you have an infection in the brain. So, congratulations. Any microorganisms in the PNS or CNS is deviation from a healthy state, and that means you have an infection. And because there is no immune system in the brain, any organism that gets in there can live and cause problems. So it's important to keep it a sterile site. So, like I mentioned before, first thing we'll talk about is the meningitises. These are the more common type of brain infections. Essentially the inflammation of the meninges, so the connective tissue surrounding the brain. So you can imagine if you have a nice thin layer of connective tissue completely surrounding the brain, if any of it inflames, it's going to put pressure directly on the brain, especially if there's nowhere for it to go outwards, thanks to the skull. Many different microorganisms can cause an infection, more serious forms caused by bacteria. You'll see this as a common theme with all of these diseases. Bacterial forms are by far the most serious. And the reason for this is most viral infections are self-limiting, meaning there's only so many cells they can infect before they run out and they stops their life cycle. Bacteria, on the other hand, they grow, they divide, they do not require a cell to survive, at least most of them. So they just grow until you're dead. The problem with the blood-brain barrier is most drugs do not pass it. So most antibiotics do not go through the blood-brain barrier, so if you have a bacterial infection, there's only a handful of drugs you can use. And not all of these drugs work very effectively. So you run into some problems that way. So if it is suspected, a lumbar puncture is performed. So that's a spinal tap looking for cerebral spinal fluid. And typical symptoms, headache, painful or stiff neck, fever, and usually an increased number of white blood cells in the CSF. Increased number of white blood cells in the CSF is a hallmark of a viral meningitis or encephalitis. So if you see a lot of that, it tells you probably have viral encephalitis or meningitis. If you can culture something out of the cerebral spinal fluid, then you have a bacterial meningitis or encephalitis. And that is because it's culturable. It's living. It can divide on its own. As you can see, headache, painful or stiff neck, fever, they're pretty nondescript symptoms. If you get these, most of the time you're like, I don't know, it's just a headache. I'm stressed or something. It's when you get these for, months on end, never relents, and then eventually it gets worse and worse and worse until you're in a coma. That's when it becomes an issue. But you'll see this kind of trend with all of these diseases where most of the early symptoms are so nondescript, no one catches them until it's too late. So that's the scariest part about a lot of these guys. All right, so first up, Neisseria meningitidis. It's It's a gram-negative diplococci, commonly known as meningococcus. So meningococcus, as you can guess, it infects the meninges, so meningitis. Often associated with epidemic forms of meningitis. The most common epidemic form of this is in college campuses, especially in dorms. The good news is there's a very effective vaccine against it. So now I think it's required, at least when I was in college in Florida, it was required that all incoming freshmen had to get the vaccine. The scariest thing about this is it's transmitted very, very easily, primarily through saliva, close salival contact, kissing, sharing food, things like that. As in very easily makes its way through the cribriform plate into the brain, where it can then infect the meninges. It's scary. And what's even scarier is that 10% of the population carry it in their nasal cavity as a commensal. So you could be making out with someone, have a little post-nasal drip, get a little bit of it in your mouth, which is gross anyways, and then you get meningitis. So it causes the most serious form of acute meningitis. So it happens very quickly, very quick onset. We're talking a matter of a few days. So you get a fever, stiff neck, and if you only get that for a couple days, you might not think anything of it until you are having a seizure or you're in a coma or something. Although the fatality rate isn't that great, it's only about 3%. And it's still pretty high, but it's pretty low considering it's a brain infection. But the good news is that there are decent vaccines against this. It can cause respiratory illnesses as well, so like a more mild or flu-like illness. And if that's left untreated for a long period of time, it can make its way into the brain as well. So, dissemination of a meningitis infection. Basically, goes through the mouth, into the sinus cavities, through the cribriform plate, where it then circulates around the cerebral spinal fluid until it basically coats all of the meninges. So once it makes its way into the CNS, hitches a ride in the cerebral spinal fluid, which basically bathes all of the meninges throughout the body. So you can see how as it divides, it's got a free ride almost everywhere. So it can be a pretty nasty, serious infection to get. How to diagnose? So it's a gram-negative diplococci from CSF sample. So if you find any bacteria in a CSF sample, you're sick. You do a quick gram stain on it if there's enough of the organism, and it's a gram-negative diplococci, treat for Neisseria meningitidis. It's as simple as that. It's somewhat specialized in the fact that it's a fairly fastidious organism, so it grows pretty slow. And it's intracellular, meaning it likes to be inside of the cell. That's how it gets through the blood-brain barrier. That's how it gets through the cribriform plate. So basically, it just burrows through the cells that are lining the blood-brain barrier. So for that reason, it's somewhat difficult to grow. And that's why generally it's grown on a chocolate agar. Since it's fastidious and it's growing outside of the cell, it needs something very, very nutrient-rich, and chocolate agar is basically oxidized sheep red blood cells. So it gets you the brown appearance. And it's also positive for oxidase. Streptococcus pneumoniae, so referred to as pneumococcus. Most frequent cause of community-acquired meningitis. So before Neisseria meningitidis, it's the most common form of epidemic meningitis. This is the most common form of community-acquired meningitis. Community-acquired means anything outside of the hospital. Epidemic means in like a close area, so in a small location, like a college campus or a small town. Very severe. 50% of the cases result in death. So it's pretty nasty. Also results in pretty nasty neurological sequelae if you survive. So 50% survive, but out of those 50%, the vast majority of them have something wrong. They lost mental capabilities, coma, seizures, whatever. Biggest difference between this and Neisseria meningitidis, at least on for diagnostic purposes, is Neisseria meningitidis causes petechia. So I think I mentioned maybe a week ago what petechia is, but just to refresh your memory, it's basically burst capillaries close to the skin. So you'll get something that looks kind of like little spider webs that form all over the skin. For Neisseria meningitidis, you get it kind of in the throat, shoulder region. For Streptococcus pneumoniae, you don't get this. So it's the biggest tall tale symptom that it's not Neisseria meningitidis, instead it's Streptococcus pneumoniae. Main reason you want to know this pretty early on is A, they require different antibiotics, and B, this one is much, much more deadly. So you want to get it as quick as possible. It's a small gram-positive flattened coccus that appears to be ended in pairs, kind of stuck together. Haemophilus influenzae. So it's a tiny gram-negative plumorphic rod. So by plumorphic, it means it can go from rods to cocci. It can change its shape depending on the environment. Its name, Haemophilus influenzae. The influenzae comes from the fact that initially they thought it was the causative organism for influenza or flu. And that is because most of the time it causes a very similar illness to the flu. So respiratory, fever, cough, malaise, nothing too serious. But if that's left untreated for a prolonged period of time, the organism can actually make its way through the pulmonary system into the cardiovascular system, which puts it right near the brain and the blood-brain barrier, or can then burrow through the blood-brain barrier into the meninges. So, biggest take-home message from this is if you have a flu for a long period of time, longer than most flus take, probably should go to the doctor and see if you have this and see if you need antibiotics to get rid of it. Causes severe meningitis, again about a 50 to 60% mortality rate. It's pretty nasty. Symptoms, fever, stiff neck, vomiting, and neurological impairment. So again, symptoms are pretty much the same for all of these guys. Causes nasty stiff neck, headache, and then eventually neurological sequelae. Listeria monocytogenes, it's a gram-positive bacteria. Ranges in morphology from coccobacilli to long filaments. Resistant to cold, heat, salt, pH extremes, and bile. It's pretty easy to find in the environment. Also, it makes its way through the gastrointestinal tract without any problems. In normal adults, causes a mild infection. In immunocompromised, that's when you get meningitis from this. So generally, it causes mild gastrointestinal upset. If you're immunocompromised, it actually can burrow through the small intestine, making its way into the cardiovascular system, into the blood, or can then travel to the blood-brain barrier, go through the blood-brain barrier, into the meninges. Pretty nasty stuff. So immunocompromised being people who have HIV/AIDS, people with organ transplants, or the extremely old, extremely young. And also, if you're a mother, you become infected, or an expecting mother, you become infected, your fetus becomes susceptible to the meningitis. Pretty nasty stuff, like most of these guys. Cryptococcus neoformans, the fungus. The fungus only capable of causing meningitis in immunocompromised patients. So you see this a lot in HIV/AIDS patients. More chronic form of meningitis. So most meningitises or encephalitises or brain infecting organisms tend to be fairly acute or quick. They don't hang around for long periods of time. This guy has the ability of kind of prolonging its lifespan. So hanging around, growing slowly, causing very, very nasty infections. Headache, most common symptom, but also nausea and stiff neck, immunocompromised, spherical to ovoid shape, and a large capsule. Again, this starts off in the pulmonary system. So it starts off in the lungs, then disseminates through the blood into the blood-brain barrier, where it then can cause infection. Now, as it's disseminating, so as it's going through the blood, the circulatory system, it can kind of set up shop all over, including the skin. So a pretty serious Cryptococcus infection basically just starts taking over everything in the body. So it's pretty nasty stuff. The good news is if you have an intact immune system, your immune system gets it right away. It never makes it to this step. If you're immunocompromised, it can be pretty nasty stuff. When you're at this stage, the only thing to do is just pump in tons of amphotericin B. I think we talked about amphotericin B slightly maybe a month or so ago, but it's basically an antifungal that if taken orally, it's pretty harmless, but if you give it intravenously, which this requires, it is the worst drug imaginable. I mean, it's just so cytotoxic, it's excruciating. But unfortunately, that's the only option. So you end up with this kind of stuff, and you're taking awful drugs that make you sick. It's just not a good experience. Coccidioides immitis. So it's an interesting fungi or mold. It has a filamentous form at 25 degrees, which is ambient temperature or room temperature. And at 37 degrees, it becomes a yeast. So it's what we call dimorphic. So it has the ability of forming these nice, large, multicellular kind of organisms, like a mold, at room temperature. And then as soon as it gets into the body, it becomes a yeast and becomes single cellular, which makes it easier to kind of work its way around the immune system and around the body. It's primarily found in the desert regions. So it has these very tough spores that can last for hundreds and hundreds and hundreds of years in very dry, sandy conditions. And then as soon as a nice big heavy rain comes, which often happens in the desert once every year or so, all of these guys, these spores open up and they form these nice big mold balls. And then all you need is to disturb the soil a little bit, breathe in these spores, and you become infected. Now this is one of the few fungi that can infect immunocompetent people. So people who have excellent immune systems can still become infected with this, and it can be pretty nasty stuff. Again, it starts off as a pulmonary infection, so it starts off in the lungs, it disseminates to the brain. Pretty nasty guy to deal with. It's got a pretty high mortality rate, about 40%. It's primarily found in the Four Corners region of the United States and in Mexico. Northern part of Mexico. Generally, the only time you see this or outbreaks of this is when there's a large rain followed by some kind of traumatic soil upbringing event, like an earthquake or a sandstorm or something. So that's when your large rain happens, allows the spores to sporulate, producing the mold, and then you have an earthquake that breaks them all up and shoots them into the air so everyone can breathe it in. But generally, you don't see too many of these on a regular basis, often because most of the regions here, with the exception of Phoenix, aren't very populated. So you don't hear about too many infections just because there's not many people there. Viruses. So everything we talked about so far have been either bacteria or fungi, so living organisms. Now we'll talk about viruses. These are generally grouped together into one big kind of infection called aseptic meningitis. It's aseptic because on culture of the cerebral spinal fluid, you get no bacteria, you get no colonies, but the person still has inflammation of the meninges. Majority of cases occur in children. It's actually pretty common in children, more so than you would ever think. But most of the time it presents itself as just a very mild flu-like infection. It has kind of a headache associated with it, or a stiff neck. 90% of cases are caused by enteroviruses. Mortality is less than 1%. So they're acute, and they're self-limiting. So acute means quick, self-limiting means there's only so many cells to infect before they stop. They run out. That's why the mortality rate is so low, because it just can't keep sustaining itself for long periods of time. Generally milder than bacterial or fungal meningitis. So most of the people, like I said, who get infected with these really don't have any major problems associated with it after, which is good. So here's a list of what we just talked about. So this will be useful if you want to study later for the exam. Neonatal meningitis. So it's transmitted in utero or during passage through the birth canal. So mom's vagina has a lot of bacteria in it, most of which are lactobacilli, which is good. But there are some other organisms in there. Occasionally E. coli, and occasionally Streptococcus aeruginosa. Generally, most moms don't have too much of this because their lactobacilli take up most of the space. But if there's something wrong, like a yeast infection or some other kind of infection that gets rid of the lactobacilli, these guys can kind of fill in the voids. When they fill in the voids, if you have the baby and they're present, they can get on the baby during birth. Birth is very traumatic, comes into contact with all the fluids from the mom, and they can actually make their way into the meninges of the baby. Babies have hardly any immune system, at least early on, so it becomes very easy for these guys to make their way to the brain. That's why things like E. coli can actually cause meningitis at this point. Because typically, E. coli would never cause meningitis in someone who has an intact immune system. But you don't see this too often. That's primarily because doctors are aware of this happening, and they'll either give you a C-section or give you antibiotics during birth if you're at high risk for this. Meningoencephalitis. So meningoencephalitis is when you have meningitis, so inflammation of the meninges caused by an infection, followed by the infection disseminating into the brain, which is called encephalitis, so inflammation of the neuronal tissue. So 2 microorganisms cause meningoencephalitis. Both are amoebas. So amoebas are eukaryotic organisms, making them very, very hard to treat. And the problem with both of these guys is they're fairly fast acting, meaning once they get in there, they go to town, and they don't stop until you're dead. First one, Naegleria floweri, pretty nasty guys, a 99% mortality rate. So I think there's only been like 2 people who have ever survived from it. Anthiomoeba, similar to Naegleria floweri. Mortality rate's not as high, it's probably about 80%, but it's still pretty damn high. Difference between the two, Anthiomoeba causes granulomas in the brain, and it's slower acting. Naegleria floweri, you got about a week or 2 before you're dead. Anthiomoeba can take a few months. So that's why the mortality rate's a little lower. It's easier to spot, easier to treat because it's slower growing. Naegleria floweri, so it's a small flash-shaped amoeba, likes to live in warm, fresh water. Wasn't discovered, I think, until the 60s or 70s. So it's a pretty new infection. For that reason, there's only been about 300 or 400 cases, just because it's only been known for 40 years or so. Likes living in warm, freshwater places, so you'll see this a lot in the south, especially in artificial reservoirs, especially if that artificial reservoir has a power plant associated with it. Power plants generate heat. That heat gets pushed back into the fresh water, warming the body with water, allowing this organism to grow. It makes its way into the brain by being forced into the sinus cavities while you're swimming in the water, and then munching through the cribriform plate. So the cribriform plate is kind of unique in the fact that it's got a very thin blood-brain barrier associated with it, and that's because there are nerves that innervate that region, primarily so that you can smell, so sensory nerves. So Naegleria floweri can actually attach onto these nerves, munch its way through the nerves until it makes its way into the brain. Once it gets into the brain, it literally just eats everything it sees. So you'll just end up with these big holes in the neuronal tissue. It's pretty nasty stuff. And it's actually interesting that within the last, 10, 15 years, neti pots become really popular. I'm sure most of you know what it is, that little pot of water, you put salt water in it and pour it down your nose. Well, there have been more cases of Naegleria floweri from people who've used neti pots because they're using water that hasn't been sterilized properly. Either they're getting it from like a stream or a lake, or unfortunately the chlorine isn't very good at killing the organism, so if it's not filtered out, it can still be present. So they recommend if you use a neti pot to boil the water first, just to make sure you get rid of all organisms in it. Because if you don't, you're basically just giving it a free ride onto the cryptiform plate and into the brain. It's not a very common infection, primarily because it needs to kind of be a perfect storm for the infection to occur. Meaning you have to be in the warm body of water where the organism is. You need to get a whole bunch of water up your nose, and it needs to be forced onto the cryptiform plate before infection can occur. But advancement is very rapid, and treatments are often futile. That's because the only way the treatment works is if you treat early on, before it has time to make its way into the brain completely. And unfortunately, the early symptoms are very mild. Headache, malaise, and occasionally fever. All of these things you would never associate with an amoeba munching on your brain. So that's the biggest problem with treatment, is it's hard to diagnose early on. And it causes primary amoebic meningoencephalitis, or PAM. This is what it does. So here's Naegleria, and these big holes are where it's been eating. Normal neuronal tissue should look like this. It's very dense, as you can imagine. There's a lot of nerves in the brain. So whenever you have holes in the brain, something's really, really, really wrong. And it just keeps going and going and going until there's nothing left.
How would you test for this one? Would it still be the spinal tap?
No, this would be a brain biopsy. So you would actually have to drill into the brain and pull out. Usually what they do is they'll go into the brain and they'll take out a long section throughout the middle of the brain and try to get all different parts and look for it under a microscope.
So it's not the kind of test you would do, it's just...
The problem with all of these guys, all of these infections we're talking about today, they like to infect the brain, and if their very first portal of entry is the brain, there's no antibodies produced because there's no immune system in the brain. So there's no easy tests, no serological tests, there's no way to look for them. And that's where it becomes a problem. So usually the best option is going for a lumbar puncture or brain biopsy. Or if you have swelling of the brain, clearly you have an infection or something. Anktioamoeba. So it's a large amoeboid trophozoite with spiny pseudopods and a double-walled cyst. Invades broken skin, the conjunctiva, and occasionally the lungs, and urogenital epithelial. So this guy can kind of get through the skin, especially through broken skin, or very thin skin associated with mucous membranes in the genitalia. And then it essentially hitches a ride in the cardiovascular system, making its way into the brain. It causes granulomatous amoebic meningoencephalitis, or GAM. Granulomatous are these large granulomas that form in the brain because of it. And this is because it moves a little slower than Naegleria floweri. It allows for the few immune cells in the brain, the microglia and the astrocytes, to kind of try to wall off the organism. And as they're doing this, they cause granulomas. So you can actually see this on an MRI. But by the time you have this, it's very, very hard to treat. Because what the astrocytes and microglia are trying to do is wall off the organism so it doesn't spread. What they're also doing is walling off the organism to treatment. So it becomes very hard to get the treatment into these granulomas. Not to mention it's hard to get the treatment through the blood-brain barrier. So you have two big major hurdles, makes it almost impossible to treat at that stage. Acute encephalitis. So acute encephalitis is by far the most common form of encephalitis. Very quick, pretty mild, usually caused by viruses. And signs and symptoms vary, but may include behavioral changes, confusion, decreased consciousness, seizures, and then the fever, malaise, headache. Very generalized, nondescript. Arboviruses. So arboviruses are the most common form of virus that causes acute encephalitis, and they're transmitted via mosquitoes. Bloodborne. So it's a little scary in the fact that a lot of these encephalitises actually occur in the United States and endemic to the United States. But the good news is that we have pretty good mosquito control and keeps them in check. But the possibility is always there. So just a few quick examples of some arboviruses. Western equine encephalitis. So it appears first in horses, then in humans. So the biggest telltale symptom that there's an epidemic of Western equine encephalitis is when a bunch of horses die for no reason. Kind of like with West Nile virus. You guys remember that little panic that happened, I don't know, early 2000s? All these birds were falling out of the sky, and they're like, oh my God, what's going on? That was the telltale symptom for West Nile virus. Telltale symptom for both of these, Western and Eastern equine encephalitis, are dead horses. It kills horses very, very easily. They're the primary target. It's carried by mosquitoes and extremely dangerous to infants and small children. So the good news is, kills horses, doesn't kill us very well. If you have an intact immune system, it's generally just a mild infection. You probably wouldn't even know you had it or that you had anything that was infecting the brain. It just feels like a flu. But if you're a young infant or small child who has a weakened immune system, it's when it becomes more serious. Eastern equine encephalitis, so east coast of North America and Canada, usually appears first in horses and caged birds, and very high case fatality rate. This one, on the other hand, a little bit more severe. Generally, fatality rates, you're looking at around 40 to 50%, which is pretty high for a viral encephalitis. Most of them, mortality rates are in the single digits. So it's pretty nasty stuff. Don't see it too often. There are random epidemics of it, but they're usually caught and kept in check. There's also a vaccine for the horses. So the thing about having horses as the primary host is horses are huge, and horses produce a lot of blood. So there's always going to be a lot of mosquitoes biting horses. So it acts as a great amplifying host, or a host that increases the amount of viral production so it's easily dispersed throughout the region. But the good news is that for Eastern equine encephalitis, there is a vaccine for horses, and I think all horses get it, at least from what I remember. California encephalitis, so may be caused by two different viral strains, California strain and the La Crosse strain. It's primarily children living in rural areas, and that's because they're the ones exposed to most of the mosquitoes. In the cities, not nearly as common. Not serious in the fact that I think in the last 50 or 60 years there have only been like 50 or 60 cases total in the United States. So it's not a huge epidemic, but it does occur. St. Louis encephalitis, or SLE. SLE is by far the most common arbovirus in the United States. Epidemic in the U.S. occurs most often in the Midwest and the South, not only in St. Louis, unfortunately. It does happen a lot in Ohio. You'll see a lot of, I think Ohio has like second or third largest amount of cases. The good news is that most of the cases are mild. Most of them are flu-like. There's only a handful that actually result in serious neurological issues. Next, West Nile virus. So most people have heard of this before, hopefully. First seen in the US in 1999. So it's endemic to the region of the West Nile Delta, which is in Egypt. Not really sure how it got to the United States. A lot of people think it was migratory patterns of birds. Who knows? Could also be mosquitoes getting trapped in planes and carried from one place to another. We don't know. 2008, 3 million people with evidence of past or present infection. So about 1% of the population in the United States. Out of all those people, only, I think it was like 0.5% of them actually died from it. And it was the extremely old or the extremely young. So generally speaking, if you have a normal immune system, it causes flu-like, mild illness, most of the time doesn't cause any symptoms at all. You don't even know you have it. So it's not very serious, except for the extremely young or the extremely old. Herpes simplex virus. So herpes primarily infects the peripheral nervous system, but occasionally it can make its way into the central nervous system. When it makes its way into the central nervous system, it becomes a hell of an encephalitis. 90% more fatality rate. Pretty nasty stuff. You don't see it very often. It's very, very rare. And if you do see it, it's generally in infants right after delivery. So from mother to child transmission, vertical transmission. Subacute encephalitis. The symptoms take longer to show up and are less striking. Most common cause, toxoplasma. Toxoplasma is a parasitic infection, primarily found in cats, but can also be taken up by humans. You also see it with measles infection, most often associated with the panencephalitis we talked about, I guess, when was that? Last week. And then lastly, prions. So toxoplasmosis gondii, subacute encephalitis. Again, it's a normal, actually very normal parasite found in cats. I think almost all domesticated cats have it. So if you have a cat, you're constantly exposed with it, unfortunately. It's a flagellated protist that basically gets excreted in cat feces. So every time you clean the litter box, you come into contact with it. It's an obligate parasite, meaning it needs to be in a host, whether that's a cat or a human. However, humans are not its primary host. In fact, it's something we call a dead-end host. So it cannot continue its life cycle in a human. But it can still cause a hell of a problem in the humans because it still thinks it's continuing its life cycle. So it will reproduce, it will produce larvae, but instead of the larvae being excreted in the feces, they get dispersed throughout the body, including the brain. And that's when you can run into subacute encephalitis. But the good news is, we, at least most of us, at least as long as you're immunocompetent, can get rid of this organism very, very quickly. So we're constantly bombarded with it, but as long as you have a competent immune system, you're fine. Unless you're pregnant. If you're pregnant, the organism has the ability of crossing the placenta and infecting the fetus, who has a very weak immune system at that point. And once it infects the fetus, it does a whole bunch of damage. So that's why, if you're pregnant, you should never clean kitty litter. Measles, so subacute sclerosing panencephalitis, or SSPE. It appears years after initial measles infections, decades. Direct viral invasion of the neuronal tissue. So it's the measles virus has kind of gone latent in kind of a weird way. And then it has this ability of making its way into the brain, causing massive amounts of virus being produced, which results in large amounts of inflammation and usually coma. Prions. So transmissible spongiform encephalopathies. Essentially what these are contagious proteins, which is kind of interesting. They're mutated forms of proteins that are transmissible from one person to another. And when this protein comes into a cell, it actually forces the cell to produce more of the mutated protein. And when these mutated proteins build up, they form big sheets. So what ends up happening in the brain is if this is a neuronal cell, which looks absolutely nothing like a neuronal cell, but I suck at drawing, and you get a prion into it, let's say the prion looks like this, it basically makes the cell produce a whole bunch of these prions. It takes over the cell. And these prions stick together in these nice long sheets called beta sheets. And they become so large that eventually they break the cell open. When you break a cell open, the cell dies. And when you start killing cells in the brain, you start running into a whole bunch of problems. Most people have heard of mad cow disease, right? Mad cow disease is a prion in cows that can actually cause the cow to become demented, so it loses its mental capabilities, loses its functions, and then eventually dies. When this is transmitted to humans, it's called Creutzfeldt-Jakob disease, or CGD, or CJD. And the only way you can come into contact with this is if you come into contact with raw brain from a cow that's been infected. So it is a protein, it can be denatured, so as long as you cook the brain, you should be okay. I generally wouldn't recommend it. The big outbreak that happened in the UK, I think it was at the end of the late 90s maybe. The big outbreak that occurred there was because they were using contaminated equipment. So equipment that had brain matter on it to make processed meat products. It's very common in the UK. They like eating gross meat all the time. Like in Scotland, they love that haggis crap, which is just everything we would never touch and just throw them together. Hopefully no one here is from Scotland. No offense, but it just looks gross. I've never tried it. But it's pretty common, at least in the UK, to take offal and mix it in with their meat products. Offal are just the byproducts of cow slaughter, including the brain. But since that big outbreak occurred, it's basically become illegal to do this. So now any brain or neurological material on the cow has to be taken out in a separate area, has to be discarded, usually by burning, just to prevent the spread of the disease. Gressman-Strauser-Strinker disease. I don't know who comes up with these names for these prion disease, but it's pretty fun. This is essentially a slightly genetic disease. So you have to have a mutation in a gene that actually allows for this prion to occur. And then you come into contact with the prion in the environment, which is pretty ubiquitous. Since most of us don't have this genetic mutation, it doesn't really affect us. And it causes a very similar disease to Creutzfeldt-Jakob disease. And then lastly, my favorite, fatal familial insomnia is only genetic. So you have to inherit it from your parents, the mutation. And basically what it does is it causes a prion to be developed in the brain. And what this prion does is just slowly but surely prevents you from going to sleep until you can't sleep at all. Like it's physically impossible. Like sleep, nothing works. Sleeping pills, nothing. So it eventually kills you because you can't sleep. So you'll be up for weeks at a time until you die. If you don't sleep, you will die. It's one of the problems being human. So, very interesting. These two, well, actually all three of them you don't see very often. These two you almost never see because it's a very rare mutation. These guys you only see in places that have poor hygiene, especially when it comes to slaughtering cattle and so forth. So spongiform encephalopathy basically causes these huge open holes. These open holes are from these big beta sheets that are formed from the prions busting open the cells, the neurons. Rabies, so it's a progressive zoonotic disease, only transmissible between mammals. So birds can't get it, lizards can't get it, fish can't get it, only mammals. Big thing with rabies. Characterized with fatal encephalitis. The way that rabies works is you generally get bitten on an extremity, your arms or your legs, that infects the peripheral nerves and works its way up the peripheral nerve until it reaches the central nervous system. Once it's in the central nervous system, it infects specific portions of the brain that actually cause you to do one of two things. Become aggressive and be scared of water. So it actually changes the way that you think and perceive things. The reason it has you become aggressive is because it's transmitted via saliva. So it wants you to be aggressive so that you bite people and transmit it. It makes you hydrophobic or scared of water because if you drink water, you'll dilute the virus in the saliva, making you less infectious. So it actually is kind of a cool virus because it's the only one that I know of that actually purposely targets specific regions of your brain to make you do different things to carry on its life cycle. So that's why whenever you see any of these kind of weird zombie movies and stuff, if it's a movie that has any kind of scientific credibility, it always starts off with a mutated form of rabies or rabies-like virus. That's because rabies is kind of like a zombie-forming virus. It does change the way you perceive things. It makes you kind of a drone. Starts off with the prodromal phase, which begins with fever, nausea, vomiting, headache, fatigue, and other nonspecific symptoms. And treatment involves both passive and active immunotherapy. So active immunotherapy, there's an active vaccine out there that we give to dogs. But most veterinarians also get it as well. And this protects them by producing antibodies. However, the passive vaccine is by far the most common, and this is because it's only given when someone comes into contact with a potential infected animal or gets bitten by an animal of unknown origin. As soon as that happens, you get the passive vaccine, which is essentially antibiotics to rabies. Or not antibiotics, antibodies to rabies. And these antibodies work to bind to the organism, preventing it from moving its way through the peripheral nervous system. Once it gets through the peripheral nervous system into the central nervous system, you're pretty much SOL. However, there have been a few cases where people have survived through this, and the only way that it's possible is by inducing someone into a coma, so giving them drugs, putting them into a coma, and then pumping them full of antiretroviral or antiviral drugs. So many antiviral drugs that if they weren't in a coma, it would kill them. And you do this for a couple weeks. Stop it, bring them back out of the coma, and then see if they're cured. If they're not, they die. However, I think there's been like 5 cases that have survived since they started the treatment like 20 years ago. So it's an option. But generally speaking, you want to catch it when it's in the peripheral state. If it's in the peripheral state, it's easy to treat. Give the passive vaccine, and you're fine. Rabies, so there's 2 different versions, ferocious and dumb. Ferocious is about 70% of the cases, and this is where you become aggressive. This is the version rabies wants. So the more aggressive you are, the more people you'll bite, the more saliva they'll come into contact with. The dumb rabies makes you hyperactive, or paralyzed, disoriented, or stuporous. So it makes you dumb, for all intents and purposes. This happens in about 30% of the cases, and both form progress to coma phase, resulting in death. How is it transmitted? So at least in the United States, it's primarily through wild animals. That's because we have a large rabies vaccination program for domesticated dogs and occasionally domesticated cats, so you don't really see it too often in domesticated dogs. However, you will see it in raccoons, skunks, foxes, and bats. However, bats aren't listed on here, and I don't know why. Bats are by far the scariest wild animal to come into contact because most of them have rabies. They don't exhibit symptoms. Also, they can transmit rabies without biting, just by coming into close contact with their saliva. So, moral of the story, stay away from bats. You see a bat, you come into contact with a bat, I guarantee you your doctor will make you get a rabies vaccine. It's we don't really know how or why it's transmissible so easily with bats, but it is. It always reminds me of an Office episode. I don't know if anyone watches The Office, where Meredith they're trying to catch a bat, and one of the guys traps the bat on top of Meredith's head, and she's like freaking out. And every time I saw that, I'm like, oh gosh, she needs a rabies vaccine. And she actually gets rabies like 5 episodes later from it. It's funny. It's funny. Even though someone had rabies. But she gets treated and she's fine. So that's why you can learn. Okay. Poliomyelitis. Polio, so it's acute enterovirus infection. Most often associated with gastrointestinal problems. So most polio infections cause either no symptoms at all or gastrointestinal problems. So diarrhea, nausea, crazy. And a small percentage of these infections will actually make its way into the spine, into the central nervous system, and cause paralysis. But it's only in about 1 to 2% of all polio infections. But the scary thing about polio, at least about 67 years ago, is that it was almost ubiquitous. It was found everywhere. Very hard to treat. Get rid of it. So, when almost everyone becomes infected, you now have a population of, I don't know, what was it, like 80, 90 million at the time? And 1 to 2% of those were becoming paralyzed. So it's a pretty significant amount of the population that were becoming paralyzed, including one of our presidents. So because of all this, because it was pretty serious, there's a huge vaccine campaign that was started, and they came out with a pretty good vaccine, worked extremely well, and now you see polio hardly at all, at least in first world countries. Third world countries, you see it pretty commonly, which is a little scary, especially in areas like India. It's still pretty prevalent. But since it is primarily gastrointestinal bug, its transmission route is fecal-oral. So as long as you have pretty good sanitation measures, you should be okay. But again, it is pretty easy for it to slip through even excellent sanitation measures. There are 2 vaccines, the Salk and the Sabin. So Salk is inactivated in the sense that it's killed and it's injected in the arm. Sabin is attenuated, so it's still alive and it's taken orally. Salk is the one that was most commonly given in the United States. It worked pretty well, but because it's being introduced into the arm instead of into the gastrointestinal tract, like it should naturally infect, it doesn't work as well. The attenuated version works extremely well because it is administered in its normal route, the gastrointestinal tract. However, it does have more side effects, and there's a slight chance that you can become infected with polio after taking a vaccine. So here's a little graph representing how many cases were asymptomatic. So you can see the vast majority of cases were asymptomatic. Didn't even know you had it. Then you get a small little percentage that cause meningitis, encephalitis, or gastrointestinal issues. Tetanus, also known as lockjaw, Clostridium tetani, gram-positive, spore-forming rod, releases a powerful neurotoxin, tetanospasm. Essentially, what it does is it mimics a neurotransmitter. So the active toxin mimics a neurotransmitter, causing all of your neurons to fire at once, your peripheral neurons. This results in tight muscle contractions. So tight that eventually it will break your spine, resulting in death. So you'll get this very tight kind of, I don't know if they have a picture of it or not. Yeah. So you get this nice kind of coiling of the spine. And this is from all of the muscles in the back kind of seizing at once. Until eventually you break your spine. Or it causes the muscles in the diaphragm to seize at once, preventing you from breathing and suffocating. It's pretty nasty stuff. Good news is there's a vaccine against it. Most of us have had it. It's required, I think in 7th grade. Lasts for 10 years. Only way that you can get this is through a deep puncture wound. So it's caused by Clostridium tetani, which is an anaerobic organism, meaning it can only grow without oxygen. It's found ubiquitously in the environment, not in its vegetative form, but in its spore form. And that's because there's oxygen in the environment, so it can't grow. Once it's in spore form, you get a deep puncture wound into deep muscle tissue that isn't oxygenated very well. These spores can open up, become vegetative, grow a little bit, release a little bit of the toxin, and very small amounts of this toxin are deadly. So all you need is just a few organisms producing enough toxin, and that's when you run into problems. The vaccine works very good at targeting the organism, clearing it, and preventing it from getting that far. Botulism, caused by a close relative to Clostridium tetani, Clostridium botulinum. It also produces a toxin, but the toxin, instead of mimicking a neurotransmitter, it actually blocks neurotransmitters. So it stops peripheral nerve function, preventing your muscles from contracting. So it does the opposite, causes flaccidity. Eventually it makes its way into the diaphragm, stopping the muscles in the diaphragm from working, resulting in you suffocating because you can't breathe on your own. Primarily, at least historically, most of the infections were foodborne in origin. So the toxin is very heat stable, meaning that unless you heat it to a fairly high temperature, it survives. And it was found in a lot of food items. So all you have to do is ingest a little bit of the food, you get a little bit of the toxin. Now you have botulism. The foodborne version was very common, at least early on, once they started doing canning in the 20s, 30s. It's because they would can the food, but then they wouldn't sterilize it afterwards. Or if they did, they weren't sterilizing it enough. So you would have a couple of these spores hanging out in there, and then as soon as you seal the can up, you basically block out all the oxygen. So now that there's no more oxygen, it becomes a nice anaerobic environment, and these guys just grow in it like crazy. While they're growing, they produce CO2, and the CO2 causes a bulge in the can. So that's why if you go to the grocery store and you have cans that are 50% off, because they have big bulges in them, throw them away. Don't get them. So that means there's a bulge, there's something growing in there, and it could be botulism. Infant botulism, entrance of the botulinum toxin in the bloodstream, very rare. Generally, the way that this occurs is if the mom gets a little bit of the botulinum toxin, not enough to actually make her sick, but it's enough for it to transmit to the baby. And very, very, very small amounts of the baby is pretty nasty, pretty toxic. But you don't really see this very much anymore. And then wound botulism, entrance of the botulinum toxin into the bloodstream. And again, the botulinum spores can be found ubiquitously in the environment. So if you are in an area that for whatever reason is anoxic or lacks oxygen, you may have some of the organisms growing there. If you come into contact with that with an open wound, you can get the toxin in that way. So here you can see it acting as a neurotransmitter or a neurotransmitter blocker, which essentially stops peripheral nerve function. And lastly, African sleeping sickness. So it's a trypanosome. It's a small little worm-like organism. Transmitted by the tsetse fly. Primarily found in Africa. You can see over here, sub-Saharan Africa. Likes to infect white blood cells. So it's intracellular. Eventually causes CNS damage and altered behaviors. So it's called sleeping sickness because it makes you really sleepy, really lethargic. And that's the first kind of telltale symptom that you probably have African sleeping sickness. It's a very slow progressing disease, so you'll have this sleeping sickness where you're really sleepy, really lethargic for decades before it actually makes its way into the central nervous system. Once it makes its way into the central nervous system, there's really nothing else you can do. It will eventually kill you. There are a couple drugs out there that work fairly well at preventing the organism and also killing the organism. But again, you can have all the drugs you want, but in places like sub-Saharan Africa where healthcare is not exactly the best, drugs don't really get dispersed as evenly as they should be. So you get long asymptomatic periods, disease onset of symptoms. Symptoms include intermittent fever, enlarged spleen, swollen lymph nodes, and joint pain. And then central nervous system is affected with personality and behavioral changes that progress to lassitude and sleep disturbances. Lassitude is just the inability to function properly. You lose your mental capabilities. That's it. Any questions? Yeah, I got one of those really good spots, like right in the front. So one problem with when your computer can't go to sleep is that it runs out of battery really quickly. Okay. All right, so now we'll head on to diseases that affect the cardiovascular and lymphatic system. You catch my breath. So, as you can imagine with these guys, they can be pretty serious because they're infecting regions of the body that are everywhere. Cardiovascular system disseminates throughout the body, so does the lymphatic system. Also the lymphatic system is where you'll find most of the immune cells. So you're infecting the lymphatic system, you're probably infecting immune cells. Once you start killing off the cells that are supposed to get rid of you, you become a pretty nasty, hard infection to get rid of. So the cardiovascular system, heart, arteries, capillaries, and veins. So it's primarily used for pumping blood. You pump blood, carry oxygen, and also remove waste throughout the body. Lymphatic system, lymph vessels and lymph nodes. Like we talked about earlier, lymphatic vessel or lymphatic system is primarily used for removing excess water and also housing the immune system. So, cardiovascular and lymphatic system and their defenses. So the cardiovascular system in itself has a lot of immune cells associated with it. That is because it is used, just like the lymphatic system, as a mode of transportation for the immune cells. But the big problem with this is that in the cardiovascular system, especially in arterioles or arteries, and in the heart itself, blood is being pushed through at very high pressures. These high pressures prevent the immune cells from doing anything. They use it as a kind of a highway to get around the body, but they can't actually function in it because they're just moving so quickly. So for that reason, infections that occur either in the heart or in the blood itself can be pretty serious and hard to treat because there's just no way for it to stop long enough for the immune system to really fight. So blood vessels and heart, they move blood in a closed circuit, also known as circulatory system. The big key here is closed circuit. So it is closed off to the environment and it's highly protected, meaning that there are many immune cells surrounding the cardiovascular system to prevent pathogens from getting in. Works pretty well. You have to be pretty hard up, meaning you have to be either really sick or immunocompromised to get a systemic infection in the cardiovascular system. And provides tissues with oxygenated nutrients and carries away carbon dioxide and waste products. Lymphatic system, so consists of lymph vessels and lymph nodes, collects fluids, and they filter impurities, fight infectious agents. So the good thing about the lymphatic, having an infection in the lymphatic system, is that there are plenty of immune cells present, plenty of white blood cells. But that can also be a problem or a caveat, because if it's an infection that targets white blood cells or immune cells, then it becomes a smorgasbord for the virus or bacteria. And that's exactly what HIV does. Defenses. So cardiovascular system is highly protected. However, if microbes do invade, they gain access to every part of the body. And again, it's being pumped through at a fairly high pressure. So it becomes hard for the immune system to kind of do anything. Bloodstream infections are systemic infections, often with the suffix emia. So bacteremia, fungeremia, viremia. And defenses in the bloodstream, the leukocytes. So you have white blood cells, but again, it's very high pressure. It's hard to slow things down. So presence of microbes in the blood, viremia, bacteremia, or fungeremia. Most often, these are transient exposures. So because the cardiovascular system is this nice highway that's being transversed throughout your body, most viruses, bacteria, and fungi that are in the blood are only there transiently. So they're using it as a ride from one place to another. However, there are some organisms that can actually grow in the blood. So if you have an organism that's just, transiently going, from the gastrointestinal tract to the lung, and it's hitching a ride in the blood, chances are it's not going to be that serious of a systemic infection. It's not going to cause that much problems throughout your body. It might cause a lot of problems in the gut or in the lung, but it's not going to be that big of a deal in the cardiovascular system. But if you have an organism that's actively growing in the blood, so it's dividing within the cardiovascular system, that becomes a very, very, very serious infection. It becomes very hard to treat, again, because immune cells aren't able to slow down enough to fight the infection. And because it kind of gets a free ride everywhere. And as soon as enough levels are built up of the organism, it's basically throughout your body. Pretty nasty, serious infection. Also, the blood is full of nutrients. We've talked before about how bacteria especially love iron. Blood is full of hemoglobin. Hemoglobin's primary molecule is iron. So it becomes very tasty for these guys. So if you have septicemia, that means you have bacteria growing in the blood. So septicemia, growing in the blood. But if you have one of these guys, viremia, bacteremia, or fungeremia, it's just transiently transmissible through the blood, just moving from one place to another. And septic shock, excessive immune response, cytokine storm, and hemodynamic collapse. So normal biota should be none, but you will almost always find microorganisms in the blood. Primarily viruses. Viruses are pretty common in the blood. We're infected with many viruses that we don't even know we have. Cause no symptoms, but they're present. And they're usually circulating around in the blood. Cardiovascular and lymphatic diseases caused by microorganisms, so they're difficult to classify. And that's primarily because they go everywhere. It's hard to say where they started, where they want to be. It becomes hard to pull them apart. Also, almost all pathogens, if you give them a free ride into the cardiovascular system, they'll probably set up shop there and start growing. So again, it becomes hard to classify. Did this start off as a septic infection? Or is it just a transient infection that turned into a septic infection? It becomes difficult to play with. And most conditions are systemic, meaning they go through the entire body. So we'll start off with one of the more chronic cardiovascular infections, endocarditis. So endocarditis is the inflammation of the endocardium, The endocardium is basically a thin protective covering around the heart. And eventually it will make its way into the valves. That's when it becomes an issue. And it is usually caused by an organism that is very good at producing a biofilm. So you'll get a nice thick biofilm growing on these valves. And because these valves are constantly being forced with a lot of pressure, rarely do you get immune cells go into that area. So they're pretty much on their own. So these biofilms just grow and grow and grow until eventually the valve collapses. You can have acute and subacute, but by far subacute is more common. Fever, anemia, and abnormal heartbeat. Sometimes symptoms similar to heart attack. In subacute cases may have enlarged spleen. Staphylococcus aureus and Streptococcus pneumoniae are by far the most common organism. And you generally only see this in older patients. That's because it takes a very long time for these biofilms to build up. Most of the time, you can come into contact with these organisms, usually through a cut in the mouth. So you'll find Streptococcus or Staphylococcus aureus and Streptococcus pneumoniae fairly commonly in the sinus cavities and in the mouth itself. So all you need is a little cut, or go to the oral surgeon or the dentist, pull out a tooth or whatever, and you can get the organism making its way into the blood that way. Once it's in the blood, it just kind of circulates around until it makes it into the heart, and it can set up shop. So here's a healthy valve. Here's an infected valve. These vegetations or biofilms growing on them. Until eventually the valve collapses. Septicemias, so this is when you have actively multiplying organisms in the blood. So these are very serious infections. Many different bacteria and a few fungi can cause this condition. So like I mentioned before, most bacteria that are pathogens, if you give them a free ride into the blood, they will become septic. Because it's easy. It's a great place to live. It's full of oxygen. If you're an aerobic organism, it's full of blood, it's full of iron, very nutrient rich. Fever, and by fever I mean extreme fever, like 104, delirium, no idea what's going on, need to be hospitalized type fever. Patient appears very ill, maybe an altered mental state, shaking, chills, gastrointestinal symptoms, all of which stem from the fever. And the fever is caused by large amounts of cytokines being produced because the immune system is just overwhelmed. Often exhibits increased breathing rate and respiratory alkalosis and low blood pressure. And again, this is low blood pressure is caused because the heart starts pumping much more slowly so that the immune system can kind of make its way into the cardiovascular system. By slowing the pressure, it becomes easier for the immune system to migrate around and actually do something. But having low blood pressure is also a problem in itself. Plague, caused by Yersinia pestis, the gram-negative rod, rodents are the reservoir, and fleas are the biological vector. So it's primarily amplified in rodents, and fleas bite the rodents, become infected, and the fleas bite you and then infect you. So it's commonly associated with poor hygiene, so having too many rodents around. Pandemics used to be very, very common. Black Death back in, I think, the 14th century. 100 million killed in the 6th century AD, but that was in the Black Death. Killed a lot of people. I think it was like 60% of the population. And the only people who survived were the people, for whatever reason, had somewhat natural immunity to the organism. They had something unique about their immune system that allowed them to survive. So for that reason, the people that did survive ended up mating with each other, producing offspring that became more resistant to the organism, so it became less and less common as time went on. You can quarantine, so that's usually the easiest and most effective way to treat. It's just by taking the person who's infected and getting them away from the healthy population. And antibiotics work extremely well. So that's why you rarely see this disease anymore, because antibiotics nip it in the bud. However, in certain third world countries, you still run into it every now and then. A couple different manifestations. So pneumonic plague, it's a respiratory disease. Pneumonic plague is by far the most deadly. 90% fatality rate. It's also the most communicable. Pneumonic means it's in the lungs. You can cough on someone and give it to whomever. Bubonic plague, so this is where you get the bacterium injected through a flea bite. So this is where it's going through its natural life cycle. Enters the lymph and is filtered by a lymph node. Infection causes inflammation and necrosis of the node. So telltale symptom of bubonic plague is a large nodule where the lymph node is located, and this nodule is full of black pus. So it's very large. There's a picture of it back in the other slide. So there's a lot of inguinal lymph nodes here in the groin region. There's a few of them that are large, and if they were to stick a needle in here and pull it out, it'd be black, pussy, kind of sludgy material. And this is called a bubo. Fancy term. And fatality rate's not nearly as high, about 40 to 50%. Still pretty high, but it's not nearly as deadly. It's also not nearly as infectious. The only way it can be transmitted at this point is if a flea bites that person, becomes infected, and then bites another person who's uninfected. In septic or septicemic plague, when the case progresses to massive bacterial growth in the blood. This usually occurs after the pneumonic plague. And this is why the pneumonic plague is so deadly. Because it makes its way into the blood. Once it's in the blood, there's really nothing else you can do. So how it's transmitted? Primarily through rodents. I don't know why they show chipmunks and squirrels, because we don't really come into contact with them as much. But it's usually rats and mice, because they are all over, behind the scenes. They become infected, often kills them. So that's one of the first signs of plague outbreak, is if a whole bunch of mice or rodents start dying for no reason. Once these guys are infected, a flea can bite them. The flea becomes infected, making it a biological vector, and then transmit it to the human. Tularemia, also called rabbit fever. So Francisella tularensis, it's highly infectious. Meaning you come into contact with very small amounts of the organism, you're going to become infected. Transmitted through skin breaks or tick bites, but most often skin breaks, especially outdoorsy types, people who like going out in the wilderness. Spread systemically, could be used as a potential bioterrorist agent. That's because it's all you need is a very small amount for it to cause an infection. Those are usually the best bioterrorist agents. Include headache, backache, fever, chills, malaise, and weakness. Can be fatal, but responds to aminoglycosides. Can be fatal if left untreated, but as long as you treat, you should be okay. So that's one of the biggest problems with it being a bioterrorist agent, is all you need to do is take some antibiotics. So here's just some differences between the two. Lyme disease. So Lyme disease is I think it's one of the most common vector-borne diseases in the United States. Transmitted by a tick. I think it's Ixodes something or another. The causative organism is Borrelia burgdorferi, which is a spirochete. Because it's a spirochete, it's very, very effective at becoming septic. We talked about this before, I think, early on in the lecture, or in the semester. But spirochetes are spirochetes because they can get through viscous fluids. The blood is very viscous. So eating a spirochete, they can kind of corkscrew throughout the blood. So they're really good at causing bloodborne infections. Rodents are the reservoir. But rodents have kind of fallen out of favor for deer. So deer are by far the biggest reservoir now. They're actually the amplification host, or the host that kind of bumps up the organism to very large levels, making it easy for it to be infected throughout the population. Biggest telltale symptom of Lyme's disease is a bullseye rash called erythema migrans. So you get just, it literally looks like a bullseye, almost a perfect bullseye. Happens, I think, in about 70% of the cases, so it's pretty common. It has this unique ability of going through antigenic switching. So antigenic switching is where it changes the molecules on its surface. So by changing the molecules on its surface, it does two things. One, it prevents the immune system from producing memory to it, because memory is dependent on antibodies, and antibodies can only bind to the surface. If it keeps changing its surface, those antibodies no longer function. And two, occasionally, this antigenic shifting causes it to produce molecules that look like molecules your body produces. So what ends up happening is your immune system says, oh, here's Borrelia burgdorferi. I'm going to produce antibodies to it. It produces antibodies to these molecules that look like your molecules. So now you have antibodies that target Borrelia burgdorferi, but also target your own tissues. Once that happens, you get an autoimmune-like disorder. And you have that for the rest of your life. So, Borrelia burgdorferi, you can treat. You can get rid of with antibiotics. But if you've had an infection for a little bit of time, chances are you'll get this autoimmune-type reaction from it. And you'll have it for the rest of your life. It's a very kind of arthritic, somewhat painful, kind of irritating disease. So it's arthritic, cardiac, and neurological symptoms develop. So it's not exactly the best thing to have. And you can see where it's primarily endemic. So in the New England region, especially in New York. New York has the most cases for some reason. Also up here in this kind of Minnesota, Wisconsin area. But luckily for us, there aren't many cases. And it's primarily because the deer that cause the infection, for whatever reason, aren't that common in Ohio. There's one deer that acts as a very good amplification host, and I can't remember the name of it. But it's interesting. It has a very interesting life cycle, too. It takes like 2 years for the tick to become fully infected. And during these 2 years, it actually kind of engorges itself with blood, falls to the ground, and then that's when it becomes very, very infectious. After it releases the blood, like after it digests the blood, then it can go off and start biting people like crazy. It's pretty nasty, especially in the New England region. There's parts of New York where you can walk around with a white sheet. They actually do this to clear the tick. They take a broom handle, they put a white sheet on it, tie some string on it, and then walk around the ground, and the ticks are attracted to white or light-colored things. The ticks just kind of jump under the fabric, and then they pick it up and they get rid of the organism or the tick that way. But you can actually see after they do it, it's just covered with sheets of ticks. It's disgusting. But for whatever reason, ticks are very common in New York. So, moral of the story, don't go to upstate New York. Lyme's disease. So it's non-fatal, evolves into a slowly progressive syndrome that mimics neuromuscular and rheumatoid conditions. Early symptoms, rash at the site. So this is the erythema migrans. Other early symptoms, fever, headache, stiff neck, and dizziness. And second stage is cardiac or neurological symptoms. And this is decades after the initial infection. So again, this is this unique life cycle that takes 2 seasons or 2 years for it to fully hatch. You have something that's infected with the organism, the Borrelia burgdorferi. Larval tick bites it, becomes infected, then amplifies the Borrelia burgdorferi inside of the tick itself. During this period, when it's in this larval stage, it's inactivated. So this larval stage could bite someone and not transmit the Borrelia. It's not until it becomes a nymph, which is kind of in between larva and adult in tick life cycle. This is when it can actually become infectious. And when a tick is a nymph, it loves to bite. it's small and it's trying to grow, so it's trying to get nutrients. So it bites like crazy, and we often are people who get bit. And then deer kind of continue the life cycle by allowing for mating to occur. So these guys, the male and the female, or the female and the male, can only reproduce on the hair of a tick. So at this nymph stage, it bites like crazy, goes all over the place, biting humans, biting deer, biting whatever. Then eventually, it makes its way into a deer where it finds its mate. They reproduce. The mother tick or the female tick falls off after it's gorged itself with blood, and then it hatches more eggs. And the scary thing is the new larval ticks that are hatched are infected as well. So it just amplifies the infectious organism. Erythema migrans is what it looks like. Pretty nice bullseye rash. Doesn't hurt, doesn't itch, just looks like a bullseye. And this is Borrelia burgdorferi, nice spirochete. Infectious mononucleosis, caused by Epstein-Barr virus, or EBV. Most of the remainder are caused by cytomegaloviruses. However, cytomegaloviruses are often asymptomatic. So most of the time when you get mononucleosis, or mono, as we call it in the United States, it's caused by Epstein-Barr virus. Sore throat, high fever, and cervical lymphadenopathy. So very large lymph nodes in the cervical region, so in the neck. These lymph nodes become very painful. It becomes very hard to move. The biggest problem with this guy is its incubation period, 30 to 50 days. And during this incubation period, you often get prodromal symptoms, which include sleepiness and malaise. So you have 30 to 50 days where you just want to sleep all day. So that's the biggest problem with this, is how long it takes for the infection to happen. You may get gray-white exudates in the throat, skin rash, enlarged spleen and liver, sudden leukocytosis, so increase in leukocytes, and fatigue is the most common symptom. Most people get infected with EBV, show no symptoms at all. There are a few people that actually go through this whole process. So I guarantee you, if I were to test all of you, I'd say 90% of you have been infected with EBV. But how many of you have actually had mono? One person? So it's a very small percentage of the people actually get the illness or the disease associated with EBV. Hemorrhagic fever diseases. Hemorrhagic fever diseases are viral in origin, and they do exactly what their name says. They cause you to bleed out of every orifice, usually. It's pretty nasty stuff. But it's pretty good at transmitting, well, it depends. It's usually pretty good at transmitting the organism. Because if you start bleeding everywhere, blood's everywhere, someone that comes into contact with you, you just pick up the organism. Yellow fever, primarily found in Central America. Mosquitoes are biological vector, where monkeys are the amplifying host. Disrupts clotting, so it stops fibrin from working properly. Once it disrupts clotting, you basically just start leaking out of your arteries and veins. Generally speaking, the hemorrhagic side of yellow fever doesn't happen very often. Only about 5% of the cases. The rest of them just cause a pretty fever, flu-like illness, which isn't terribly severe. Dengue fever, Asia, Central America, again transmitted by mosquitoes, can be a mild infection or something called breakbone fever. It actually reduces, which is weird, the calcium in your bones, making them very brittle. Chikungunya, which is found in Africa, which is very similar to dengue, again transmitted by mosquitoes. It breaks bones by reducing the amount of calcium and causes an arthritic stance. Arthritic stance is kind of a hunched over appearance, which is from brittle bones. But generally speaking, it doesn't cause that much hemorrhagic fevers, or hemorrhaging, which is bleeding out of every orifice. But it is a possibility in about 1% of the cases that become infected. Ebola and Marburg, so primarily found in Africa. Transmission is unknown. It's thought to be transmitted via bats, but we're not exactly sure how or why, but bats do become infected. Causes severe hemorrhage. So almost all the cases of Ebola, you start bleeding out of every orifice. So the eyes, fingernails, anything that kind of has thin skin or an easy opening, you bleed out of. Mortality, 25 to 100%. It's closer to the 100% range. There are epidemic cases of this that occurred in southern Africa in the mid-70s, where it just completely obliterates tribes. So the biggest problem with Ebola is it's kind of a stupid virus. And the fact that it works too quickly and causes symptoms that are too noticeable. So what ends up happening is you get infected, and within a week, you're bleeding out of every orifice, and then you die. So if I was, if we were a tribe, and you became infected with Ebola and started bleeding out of every orifice after a week, would I go near you? No. I would stay clear away from you. And generally, you're the most infectious while you're bleeding.
I'm going to die. I don't know that way.
So what generally ends up happening is if you're a tribe in Africa, one person becomes infected or a few people become infected, the rest of the tribe leaves you and then goes to a new area and stops the transmission of Ebola. So it's kind of fizzles itself out in a way because it is so deadly and the symptoms it causes are so severe, it's very easy to stay clear of it. Lassa fever, 20% asymptomatic, can develop severe hemorrhage. Rodent droppings are how it's transmitted. Non-hemorrhagic fevers. So the biggest telltale symptom of these, high fever, but no hemorrhage. Brucellosis, Q fever, cat scratch fever, Trent's fever, HGA and HME, and Rocky Mountain spotted fever. A lot of these are transmitted by ticks. Brucellosis, so Brucella abortus, Banks disease, causes an undulant fever. No, that does not mean it's caused by cows. Undulant means it goes up and down. It's very cyclical in nature. So you have a high fever followed by a low-grade fever, high fever followed by a low-grade fever. It's very systematic. You can kind of track it that way and determine how far along the infection is. The zoonotic disease enters through breaks in the skin and causes a long-term fluctuating fever. By long-term, I mean several weeks to months. So you're constantly going up and down, almost like you have menopause when you're going through heat flashes. That's what it feels like. Q fever, Coaxiella Bertinelli. Q equals query. It's a gram-negative rod, endospore structure, transmitted to humans by inhalation, primarily from digging through soil. Sudden onset of fever, chills, and body aches. Query because they didn't really understand what was causing it at the time. Cat scratch fever. Starts 1 to 2 weeks. Bordenella henselae. Cluster of small papules at the site of infection. It's called cat scratch fever because the organism is found in cat claws. So often when a cat scratches you, you'll get an infectious cut. That's what it turns out to be. It's just like having a cut that got infected and it's all pussy. Nothing too crazy. Occasionally you can get lymphadenopathy from the draining lymph nodes. So you'll get an enlarged lymph node from wherever the scratch is drained to. And occasionally you get fever. But it's very mild, almost 0% fatality. Almost unheard of with these guys. Trench fever, so Bartonella quinitania, carried by lice, common in soldiers, especially during World War I. Digging trenches and hanging out in them for months at a time was very common. All it takes is a couple lice, and then the whole platoon is infected. Highly variable symptoms. However, a 5 to 6 day fever is almost always present, but everything else is kind of mismatch. You have fatigued, achy, just depends. Leg pains, especially in the tibial region, headache, chills, and muscle aches. Macular rash can occur, and endocrinitis can develop. So you can see all of these are kind of variable, and you don't get all of them, or only some of them. Some people get none of them. It really just depends. But the fever is almost always present. HGA and HME. So this is a tick-borne fever-producing disease. Human monocytic urticulosis, or human granuloma, and aponeuroses. Most people refer to this to ehrlichiosis. It's just a kind of generic term. Basically, it causes your endothelial cells that make up your arteries and veins to become leaky. So they lose their tight junction ability, and you start losing fluid. So you get a lot of edema. Acute febrile state, headache, muscle pain, and rigors. Rigors are tensing the muscles. Rocky Mountain spotted fever. It's caused by a rickettsial organism, Rickettsia. Gram-negative intracellular bacteria, so it's very small. It's an obligate intracellular parasite, 2 to 4 day incubation, most prevalent in southeast United States, not just in the Rocky Mountain region. So last time I checked, the southeast United States isn't the Rocky Mountain region, but that's what was first described in Colorado. It's usually associated with tick bites, but it can also occur just with open wounds, become contaminated with soil. First symptoms: sustained fever, chills, headache, and muscular pain. You get a distinctive spotty rash, where the spotted fever thing comes from. And in most untreated or severe untreated cases, enlarged lesions merge and become necrotic. So you get big, large necrotic holes in your tissue or your skin. Other manifestations: cardiovascular disruption, delirium, convulsions, tremor, and coma. The good news is that most of these infections do not result in this. Small fraction. But it is still a possibility. Malaria. So malaria is the most common parasitic infection in the world. Most common parasitic infection in the world. Primarily caused by two different flavors, Plasmodium falciparum and Plasmodium vivax. Anopheles mosquitoes are responsible for transmitting the organism. They act as the biological vector. And these mosquitoes only live in the equatorial region, so they need to be in the warm tropical region. That's why most cases are concentrated in the equatorial or the subtropic region. It's endemic equatorially, and 300 to 500 million cases at any given time. So there's what, 7 billion people in the world? It's a pretty significant amount of people are infected. 2 million fatalities every year, fought primarily by vector control and chloroquine. So the easiest way to prevent infection is to get rid of the mosquito. It's either using insecticides, or because the Anopheles mosquitoes primarily bite at night, they're nocturnal, one of the easiest and most cost-effective methods is to install a mosquito net over your bed. So it's becoming big campaigns, and especially in Africa, we've got to disperse these nets to as many people as possible, trying to prevent the spread of malaria. Chloroquine also works pretty well, especially as a preventative measure. So most people who travel to these regions from first world countries will take chloroquine for a prolonged period of time while they're in the area to prevent the infection from occurring. And chloroquine is actually a derivative of quinine. And quinine is the bitter substance put into tonic water. And it was actually developed as a malarial agent, if anyone's interested. So drink tonic water if you don't want mosquitoes to bite you. It works. Malaria, so the world's dominant protozoan disease. Very, very, very common in the equatorial regions. It is a 10 to 16 day incubation period. First symptoms are malaise, fatigue, vague aches, nausea with or without diarrhea. So kind of nondescript symptoms. Next symptoms are bouts of chills, fever, and sweating. And this fever undulates, meaning it goes up and down very regularly, very cyclically. Keep track of it. Symptoms occur at 48 to 72-hour intervals. And this is what usually tells the doctor what flavor of malaria you have. Because some flavors are easier to treat than others. Some flavors are more deadly than others. And the interval length and regularity of the symptoms reflect the type of malaria. Malaria primarily infects two areas of the body, the liver and the red blood cells. The red blood cells are where it does most of its damage. So once it infects a red blood cell, it eventually kills the red blood cell, preventing it from carrying oxygen throughout the body. So it essentially causes you to become anemic. Depending on the flavor of malaria, this anemia can occur anywhere from 1 to 2 years to 10 to 15 years. It just depends. And once you get severe enough anemia, there's a good chance you're not going to make it. So it has a fairly complex life cycle, which I'm not going to expect you to know, but I will talk about it. So it has two phases, an asexual phase and a sexual phase. The asexual phase is where it starts its infection, at least in a human. So you have a mosquito who bites a human, trying to get its blood. When a mosquito bites, it injects some of its saliva into the bite. It does this because the saliva acts as a numbing agent, so that you don't really feel it. Well, the malarial parasite hangs out in the salivary gland. So as that saliva gets injected in, the malarial parasite goes with it. Once this sporozoite, which is the larva of the malaria, get into the blood, then makes its way to the liver. Once it gets to the liver, it infects liver cells. And the primary mode of malaria in the liver cell is to produce many, 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 many copies of itself. So it acts as kind of like an internal amplifying host. So you get these sporozoites, they infect, they make many copies of themselves. Then these many copies burst out of the liver cells and become merozoites. So merozoites are many, many copies of the sporozoite. I know the names suck. It's fun, isn't it? Once the sporozoites are released from the liver, they're let go into the cardiovascular system. Here they come into contact with red blood cells. They attach to the red blood cell, grow and divide. As they're growing and dividing, they become ring trophozoites. And they give the name ring because they look like this. They cause these little ring-like structures in the red blood cells. So the easiest way to diagnose malaria is to take a sample of blood, put it on a slide. This is what it looks like. It's pretty easy to diagnose. So now you have red blood cells that have these ring trophozoites, which are again amplifying. So it acts as a secondary amplification step. These trophozoites burst out of the red blood cells. When they burst out of the red blood cells, they become gametocytes. These gametocytes can then bind to uninfected red blood cells and just hang around and wait. And they're waiting for a mosquito to bite this infected person. When the mosquito bites the infected person, the gametocytes are drawn into the mosquito's gut. Now the gametocytes, there are male and female versions of these. So when they get drawn into the gut, you now have male and female gametocytes. Once digestive enzymes inside of the mosquito's gut starts to break down these gametocytes, it allows for them to reproduce. So now you have a male and a female coming together, and you now have sexual phase. After the sexual phase, they produce sporozoites which travel to the salivary glands, lie in wait dormant to infect a new unsuspecting victim. So it's a pretty complex, crazy life cycle. And it has a lot of fun terms, so I'm not going to expect you to know that. But I do expect you to know that it infects both the liver and the red blood cells. Anthrax. So anthrax is caused by Bacillus anthracis, bacterium, spore former. So it works very well as a bioterrorist agent. That's because these spores are very hardy, being disseminated through a population. There are many different forms of anthrax: cutaneous, pulmonary, gastrointestinal, and anthrax meningitis. Cutaneous is the most mild, primarily infects the skin, causes these large kind of black necrotic lesions. Pulmonary anthrax, most severe. It's the inhalation version, so you're breathing in the spore. Once it's in the pulmonary region, it can actually make its way into the cardiovascular system. In the cardiovascular system, it then gets a free ride to the meninges via the blood-brain barrier. Gastrointestinal tract is also fairly mild in the fact that it causes pretty nasty diarrhea and nausea. And often, the organism has the ability of burrowing through the small intestine, making its way into the blood. Cutaneous and pulmonary forms are the most common, with pulmonary being the most severe. 80% mortality rate if no treatment's administered. But antibiotics work pretty well against anthrax. Oops. So Bacillus anthracis is a gram-positive rod, has animal reservoirs, primarily cattle. So it's found all over cattle. Can affect various locations. Cutaneous and pulmonary infections are most common. Polypeptide capsule. This is one of its great virulence factors. Prevents it from being cleared easily from the immune system. And has been used as a biological warfare agent. Can you tell I'm getting sick of lecturing? I'm just like, all my words are becoming words never heard of before. All right, so now my favorite, HIV. So the rest of this lecture will be about HIV and AIDS. So HIV is a retrovirus, meaning it has an RNA genome, which it then transcribes into DNA. But it's a special type of transcription called reverse transcription. That's because normal transcription is going from DNA to RNA. HIV, it's going from RNA to DNA. So it takes its RNA genome, makes DNA out of it, and then takes that DNA and incorporates it into the host genome, so the cell that it infected genome. This gives it a very long life in the infected individual, because now that cell is always going to be infected. It's very hard for the immune system to target and clear. It's a hybrid virus. So it originated with SIV, so simian influenza virus, immunodeficiency virus. Simian stands for monkeys, and it is thought to have happened in humans back in about, I think, 1940, 1950s, in the Horn of Africa. Just a little kind of curved out part in Africa. I always get this question: was it people having sex with monkeys? And I'm like, no. It's probably caused by tribes who hunt monkeys and eat them for food. They often would hunt, kill the monkey, starve its blood and all that kind of stuff. And by doing that, it's a pretty, involved process. You get blood in your hands, blood in your eyes. You can transmit the organism that way. The really unique thing about SIV and HIV is that it's highly mutable, meaning it mutates itself like crazy. So what ended up happening is the SIV mutated itself just enough to where it now can infect humans instead of monkeys, and this gave you HIV. The sad thing is that SIV in monkeys is harmless. It's like having CMV. It causes general malaise for like a couple weeks. Their immune system kicks in, gets rid of it. They have it for the rest of their life, but it doesn't really do anything to them. The HIV in humans, since we haven't really evolved very much with it, it doesn't treat us very well. Eventually, HIV results in acquired immunodeficiency syndrome, or AIDS, and this is because the cell that HIV targets are CD4 T cells. So it's a virus that targets your immune system cells. So by killing the immune system cells, it prevents your immune system from functioning. If your immune system can't function, you now open yourself up to a whole bunch of infections that you normally wouldn't get. And these infections are what eventually kill you. When you get to this state, when your immune system is that low, you now have AIDS, or acquired immunodeficiency syndrome. So it's not HIV that kills you, it's an infection from another pathogen or opportunistic pathogen that eventually kills you. Spectrum of clinical disease associated with HIV infection tends to go, is pretty unique, and we'll talk about that in a little bit. And symptoms directly tied to two things, the level of virus in the blood and the level of T cells in the blood. So the spectrum of clinical disease looks something like this. So here's day 0. This is when you come into contact with HIV. So HIV is transmitted via blood. So it's a bloodborne pathogen, primarily transmitted either via sex, intravenous drug use, or from mother to child. You become infected, and immediately what happens is the virus shoots up. So the virus has made its way into the body, into the host. It's found its CD4 T cell, it infects it. CD4 T cell produces tons of virus. The amount of virus in the blood goes up. So within a week or two, you have very, very, very high levels of HIV. Now, as you would assume, because you have such high levels of HIV in the body, the amount of CD4 T cells starts to drop. Because these viruses are infecting the cells, they're killing the cells to produce more virus. And then about 2 weeks in, your body starts to produce antibodies to HIV. And these antibodies work fairly well at killing the active virus. So as antibodies start to be produced, the virus drastically drops. So in this beginning period, from about day 0 to about 2 weeks, this is what we call the convalescent period. So this is when you get symptoms of HIV, if you get any at all. And generally, they're very nondescript, flu-like, fever, headache, malaise, things you wouldn't necessarily associate with HIV. But if you were to get symptoms, this is when it would happen. Now, because of the antibody production, because of the drop in viral production, the amount of T cells stops dying off. They stay pretty steady. And as they stay steady, the amount of antibodies stays steady and the amount of virus stays low. And this is what we call the latent period. So from about, I don't know, maybe a month or so after initial infection to 10, 15, 20 years, you're in this period of where you feel absolutely normal. Your CD4 T cells are pretty normal. They're a little low, but everything's okay. You have a little bit of virus in the blood, so you can still infect people. But all in all, you feel great. And then one day, something happens where your CD4 T cells drastically drop. The viral infection takes over, starts infecting the rest of the CD4 T cells, slowly killing them off until you have almost none at all. And once you get to less than 200 cells per microliter in blood, that's when we tell or consider you someone who has AIDS. So at this point, this is when you become susceptible to opportunistic infections, which eventually will kill you. The good news is that this period down here used to be very variable, depending on how much virus you came into contact with, or what type of immune system you have. So some people, this down here would only be 6-7 months. Some people it would be decades. Really just dependent. But thanks to the advent of antiretrovirals, which are drugs that stop HIV production, this period down here lasts for 40, 50, 60 years. It's a very manageable disease, meaning as long as you take your antiretrovirals and you go to the doctor, you live a normal life, and you become severely less infectious. You still have a little bit of virus in the blood, but it becomes very hard for it to be transmissible to others. However, you still have the disease, and there currently is no cure. The reason there's no cure is because of its unique ability to make DNA from its RNA and incorporate that into CD4 T cells. So during this process, at least early on, a lot of T cells become infected. Most of these T cells produce virus and die. But a very small percentage of these cells, about 1% of them, do not produce virus. Instead, they just hang out like regular T cells. And later on in life, somewhere down in this period, the AIDS period, for whatever reason, they wake back up, start producing virus, and the rest is history. So currently what my lab is trying to do is figure out what causes these cells to become latent and how can we either prevent it or get rid of it. The biggest problem with latently infected T cells is that antiretrovirals do nothing to them. Antiretrovirals only go after actively producing cells that are actively producing virus. So one of our current strategies is to turn on all of the latent virus and then pump in a whole bunch of antiretrovirals and hopefully kill off any infected cells. But it's just starting to make its way into clinical trial. Like, not even clinical trials, research trials. So hopefully in the next 10 years or so, it will be pretty common. So HIV/AIDS symptoms, so initial infection is vague, mononucleosis-like symptoms that soon disappear. And most people do get some sort of symptoms, but they're very nondescript. And a good 30-40% of people get absolutely no symptoms at all. They feel nothing. This is what makes it a very, very good virus. Because it can infect people without them knowing that they're infected. And then its mode of transmission is via sex, which is very common because people need to reproduce. So it makes it a very effective pathogen. Period of asymptomatic infection that varies in length from 2 to 15 years. So again, this depends on your immune system. Also depends on how much virus you came into contact and what type of HIV you came into contact with. And then initial symptoms of AIDS, fatigue, diarrhea, weight loss, and neurological changes, primarily due to the opportunistic infections that occur. Other symptoms as the disease progresses, so opportunistic infections of neoplasms. Most commonly, these are fungal in origin, and they're very hard to treat. Severe immune deregulation, hormone imbalances, metabolic disturbances. The whole body goes out of whack as soon as you start screwing with the immune system. So you get a lot of problems. Pronounced wasting of body mass. Also, some of the antiretroviral drugs can cause this. So it's kind of a double whammy. Protracted fever, fatigue, sore throat, night sweats, lesions in the brain, meninges, spinal cord, and peripheral nerves. So neurological HIV, neurological AIDS, also called HAND, which is HIV-associated neurological degeneration, is just becoming widely understood. I think most people were kind of discrediting it first. But I think most people believe it happens, and it's similar to having Alzheimer's. So, but having Alzheimer's pretty early on in life. Here's just a list of AIDS-defining illnesses. Most of these are viral, or not viral, fungal in origin, and most of them wouldn't touch us because we are immunocompetent. So it's an enveloped RNA virus, part of the retrovirus family. Carries a unique enzyme called reverse transcriptase. This is the enzyme that takes the RNA genome of HIV, reverse transcribes it into DNA, which then can be incorporated into the cellular host genome. Binds to CD4 cells. So CD4 is required for entry. That's how it targets CD4 T cells. But CD4 is also found on macrophages. It's also found in dendritic cells. So HIV can infect macrophages and dendritic cells. What do macrophages and dendritic cells do with T cells? You remember? CD4 T cells. They communicate, right? They do that by recognizing MHC, right? So they have to talk to each other. So what ends up happening is you get macrophages, dendritic cells that are infected. A T cell comes by to communicate, and the HIV viral particles get dispersed into the CD4 T cell. So they act as kind of an amplifying host within the patient. HIV replication, I'm not going to bore you with the details, but essentially RNA becomes double-stranded DNA, which gets incorporated into the host genome. Some of this RNA can also be used to produce HIV proteins, which eventually bud off of the cell. Routes of transmission: sexual intercourse, blood products, and IV drug use. Also can pass through the placenta, but very, very rarely, and breast milk. Breast milk is a big problem in areas like Africa, primarily because clean water is hard to come by. So women breastfeed because they can't, get dry formula and add water to it. So if they're infected, it's very easy to transmit it to the baby via breast milk. Sexual intercourse. So HIV is actually a pretty hard virus to become infected with, contrary to what most health classes in high school tell you. For heterosexuals, it differs between men and women, but generally for a woman, if you had sex with an infected man, you have about a 1 in 10,000 chance of becoming infected. For a man having sex with an infected woman, you have about a 1 in 100,000 chance of becoming infected. So that means I would have to have sex 100,000 times, statistically speaking, with that infected woman before I became infected. Now, this is if you're having normal sex without any breakage or bleeding or anything strange. As soon as that happens, then everything changes. But just normal, run-of-the-mill sex, heterosexual sex, it's pretty hard to get. Now, if you're having anal sex, whether homosexual sex or men and women having anal sex, becomes a little different. So if you're infected and you're giving the anal sex to someone who's uninfected, they have about a 1 in 10 chance of becoming infected. The primary reason for that is penises aren't supposed to be in the anus. So they're not designed to handle that kind of trauma. So a lot of bleeding, a lot of skin fissures, a lot of things happen, making it very easy for the virus to make its way in. Now, if there's a person giving anal sex to someone who had HIV, they have about a 1 in 100 chance of getting HIV. Because they're not going through as much trauma, because they're not having things done to them that shouldn't be done, naturally speaking. So, contrary to what most people believe, it's pretty hard to get HIV from sexual intercourse. But because sexual intercourse is so prevalent, because it happens all the time, multiple times, with many different people occasionally, it becomes a pretty common route of transmission, especially in the United States. However, it's different depending on what country you go to. In areas like Eastern Europe and Russia, intravenous drug use is by far the most common transmission for HIV. If you go to areas like Africa or certain parts of Asia, you still see blood products that are infected because they generally don't have, or sometimes they don't have the right laboratory techniques to diagnose infected blood from uninfected blood. So it really just depends on where you're at. AIDS cases in the US. So male-to-male sexual contact, so you can see it's about 46%. This is why it has a stigma of being a homosexual disease, at least early on in the epidemic. It's not because it only happens in homosexuals, it's because homosexual sex tends to be a little bit more traumatic, resulting in more blood and more chances of transmitting the virus. Injection drug use, 25, 17%, so it's gone down a little bit. And this is generally ascribed to harm reduction strategies, which are basically giving out free needles to intravenous drug users so that they don't have to reuse the same needle over and over again with their buddies. It's funny, in Australia they actually started this harm reduction strategy back in like 82 or 83, before they even knew that much about HIV. And so now, intravenous drug use in Australia is still pretty prevalent, just like here. But the amount of people that have HIV because of it almost zero. That's because they can go to any clinic, they can go to little shops that they've set up and just get free needles. I mean, if you think about it, people are going to do intravenous drug use no matter what. You might as well give them clean needles so they're not getting diseases from it too. But that will never happen in the United States, at least on a wide scale basis, because we're in the United States. Male-to-male sexual contact and injection drug use. So these guys are what we call the bridging population. So male-to-male sex contact, you can see how prevalent it is in that population. What ends up happening is you get people infected in this group, you then do intravenous drug use with their buddy, who happens to be heterosexual. So now they've just transmitted the HIV they got from having homosexual sex to a heterosexual male without having sex with them. So we call it the bridging population because it's kind of connecting the two together. Again, back to the Australia example, male-to-male sexual contact for HIV transmission is about 70% of the cases in Australia. Heterosexual, about 30%. And those are almost all caused by sexual encounters, not because of intravenous drug use. So it really kind of narrows the gap between the two. Heterosexual contact in the United States, 11, up to 31% in 1 year. So it's becoming more prevalent, especially in this bridging population, also in a population of African American men who are what quote unquote are called on the down low, which essentially means they have homosexual sex, but they still think they're heterosexual. They live heterosexual lives. But then they have things called poker parties for whatever reason, where they get together and have homosexual intercourse. But then they still think they're heterosexual, so they go back to their family, their wives, have sex with their wives, and transmit the virus. Used to be very, very common in the 90s. It's becoming less of a problem now that the stigma for being homosexual in African American populations is becoming less severe. So it's becoming more normal to be homosexual. And then other is just, who knows? Breast milk, placenta, infectious blood products. Mechanisms of antiretroviral drugs. So antiretroviral drugs significantly increase the life expectancy of someone who's infected. The latency period gets very long. They basically live very normal lives. So like I mentioned before, HIV has the ability of mutating fairly easily. So it does have the ability of mutating itself to the point where it no longer responds to the drug that you're taking. But because HIV research is such a hot button topic and it happens all the time and there's a lot of money for it, there are constantly new drugs being released. So at this point in time, most scientists and clinicians believe that you can live a fairly normal life expectancy with HIV, because even if your HIV virus becomes resistant to the current antiretroviral you're on, there will be a new one popping up that you can take and suppress it. Generally, they work in a couple of different ways. They can stop fusion, so they can stop the virus from getting into a cell, preventing infection. They can stop protease. Protease is a special enzyme that makes HIV proteins work. Without protease, the HIV proteins stay in one long strand and they don't actually become a virus. So if you block that, you stop viral production. And then integrase inhibitors. Integrase inhibitors stop the HIV double-stranded DNA from integrating with the host DNA. And for whatever reason, your book likes to leave out, oh no, here they are. Nucleoside analog or non-nucleoside analog reverse transcriptase inhibitors. These basically stop reverse transcriptase from making DNA from its RNA. So generally, the most common treatment is 2 of these NNRTIs or NRTIs and 1 protease inhibitor for the rest of your life. Used to be pretty problematic because you'd have to take it twice a day and you take 3 pills twice a day. It's kind of annoying. But they've been able to reformulate the drugs so where you can take 1 pill per day and be fine. But the biggest problem with that is that if you take any breaks whatsoever, meaning you stop taking your drugs even for a couple days, maybe a week, it allows the HIV kind of mutate itself a little bit because it starts growing. and it will or could potentially become resistant to the drug. So the key with any HIV or retroviral therapy is to take it as long as prescribed, just like antibiotics. Next, adult T-cell leukemia. So it's a leukemia that affects T cells. So leukemia just means reduction of white blood cells, and in this case we're talking about T cells. And it's very similar to HIV. However, it's not nearly as transmissible, which is good. You need very large amounts of the virus to become infected. You get adult T-cell leukemia or hairy cell leukemia. Signs and symptoms include bruising or bleeding, paleness, fatigue, recurring minor infections. Not very severe. It's usually manageable. That's it. Any questions? All right.